Okay, Akbal's here after his 2 ice creams. Everybody else get a free ice cream? I noticed 2, yeah. Did you eat 2? Oh, good, okay. I noticed it on there, it said caution children, which is probably appropriate for a university, isn't it? Okay, as some of you probably noticed, I haven't been able to release the results for report part A. We had a IT hiccup on Friday, another one again on the weekend and this morning. So we're still playing catch up on uploading because I have to be able to get on, turn it in to check the similarities, which I've been doing in between it working and not working. Hopefully tomorrow, maybe the next day. Promise Friday at the latest. Get that going. And this week, you know, you have no wet labs on this campus, no wet labs on Hawkesbury. If you're a Parramatta student, you might be in a Tuesday class, which means there's still labs going on. What you're doing instead, you don't come into the lab at all because you're going on to views and you're watching those instructional videos, right? You've been looking at the notice on views and it says to look at that particular module 5 objectives, yeah? It says to find a couple of assays, one from, who was it, Sigma was one, and the other one for the cortisol assay. You need to get those protocols because when you come into the lab next week, you are basically on your own. The demonstrator will give you instructions on when to do each step in the cortisol assay. Protein assay is all yours. You'll need to note very specifically, up on views, you've been assigned a patient number, haven't you, for the mid-semester exam? That's the same patient number you pick up when you come to the lab, is that clear? So whatever patient number you have on views, supervisor will have that same list and they'll say that's the patient number you need. You'll get 2 tubes, 1 tube for salivary protein, 1 for salivary cortisol with your patient number, is that clear? We agreed last week across all 3 campuses that at the end of the lab session, which when here is Thursday or Friday for this campus, at the end of the lab session, you have 24 hours to submit your response to views. Everyone clear on that? Notice on views. 3 questions. 1, what's the cortisol? 2, what's the protein for your patient? 3, a couple of multiple choice questions based on the assay, particularly the cortisol. That's also 1%, 0.5% each. Everyone clear on that? There's no lab book marking, there's no team leaders, they're all done. Anybody who hasn't been a team leader has a special consideration, they'll be giving their team leader presentations in front of me. That'll be fun, won't it? Oh boy, maybe you wished you'd done it before now, eh? In front of me. We are getting close to the end of the semester, aren't we? We've got this week, face-to-face, next week we're face-to-face, yep. Integration Metabolism 3, last week, week 14, whereabouts are we? Online, Blackboard, Collaborate. I'll warn you today, the last 2 slides I'm going to just briefly introduce because the concepts I need to get in your heads for this week, next week and for the review. They're a little bit complicated. We have a few more concepts to deal with today. And timely, given if you've seen the Nobel laureates who got Nobel Prize, 3 characters dealing with what? Oxygen uptake in cells. I talked about that a couple of weeks ago, HIF, didn't I? Hypoxic inhibitory factor. Why cells take up oxygen, what happens to their DNA, their behaviour particularly. So you guys know about it. Today we're moving more on to the endocrine factors, and I introduced this last week briefly, giving you some simple hormones to start with, giving you an idea of how they work via their receptors, receptor number, a little bit of signal transduction, and the response to the cell. Well, I'm afraid it just starts to get more complicated, like it always does in metabolism and cell biology, particularly in you. Now, this is you up here. You have, in the centre of your brain, a pineal gland. Hopefully you've heard a bit about that pineal gland, often called the third eye. That gland is sensitive to light. That's going to alter your circadian rhythm, your diurnal pattern we're going to talk about today. Some of the stuff I'm talking about today is not in the textbook, unfortunately, because it's relatively new. You need to keep in mind that all of your cells, more and more of them now, you want to jot down the term, have clock genes in them. Genes that regulate your diurnal rhythm. that regulate your metabolic processes to link with day length. How much light, how much dark. That's also overlaid by what you do, how much you eat. So up here in the pineal, you're going to write down clock genes. There's about 7 of them in your pineal. I'm not going to take you into all the gene jockey stuff in this particular unit. Last week we talked about the hypothalamus. There it is there, hanging down from the base of the brain. That hypothalamus takes signals from your neuronal input, from your eyes, your taste buds, from your ears, wherever, and integrates them, but also now from your pineal. Your pineal is going to do things to you to change your behaviour based on light and dark cycles. That hypothalamus hangs down. If we zoom into that, that's this little call-out box. Here's our hypothalamus. You'll see neurons coming in there where my laser is. What do they stimulate? Hypothalamic cells who turn a neuronal signal into an endocrine signal, a hormonal signal. So the hypothalamus here secretes hormones into this blood vasculature, which zooms through your pituitary, which hangs down, like I said, a bit like a punching bag, with an anterior part and a posterior, front part and a back part. The anterior does a lot of work. There's a lot of hormones developed in that anterior, stored in vesicles, releasing hormones during the daytime, during the nighttime, and during stress events, which is that final exam about what's going on. This sets up the notion, like we saw last week, every hormone has a receptor. Now, if it's a peptide-based hormone or an amino acid-based hormone, it has a cell surface receptor. How many receptors? Well, we have receptors on the pituitary. Hormone into this blood circulation, into the pituitary, we stimulate receptors. Coming from the hypothalamus, we have hypothalamic releasing factors. Write it down, releasing factors. Their job is to only go a short distance into this pituitary to release specific hormones via their receptors. When they stimulate those cells via their receptors, a signal transduction cascade, those cells here in the pituitary particularly, we're going to focus mostly on it, the anterior particularly, then we release other hormones, and there's a whole lot of them. And I'm going to focus on the ones that work to metabolism particularly. What this sets up is a hierarchy. That's a term you want to jot down, an endocrine hierarchy. We start in the brain, you integrate signals, here we are, the sensory input, photons, sounds, taste, whatever it is, talk about next semester, to the CNS. CNS produces response in the hypothalamus. What does the hypothalamus produce? Releasing factors. Write down hormones or underline it. Those releasing factors work specifically on the anterior pituitary. That's where they mostly work. Very few of them get outside of that region of the brain. When they hit the anterior, just write down receptors on the cell surface for those, because most of those are peptide-based hormones. When you reach the anterior pituitary, you get a whole lot of different hormones doing different things. Some alter your periodicity in terms of your reproductive cycles, things like follicle-stimulating hormone, luteinizing hormone. They affect your ovaries and testes. Their functional role, which means it goes from the pituitary, the anterior, all the way down to your ovaries or to your testes, regulating input. But the ones you write down, there's feedback back to the brain as well. Feedback from things like estrogen, testosterone, back to these areas. And when you get to those particular regions, you get inputs from progesterone, estradiol, testosterone. You change your reproductive organs. You change how much sperm you produce, how much testosterone, how many cycles you've got in terms of eggs. But it also changes your body, and you want to jot down, it changes your metabolism. More testosterone, you lay down more protein. More estrogen changes your behaviour as well, which we can go into in other units. A few other odd ones. Somatotropin, you're going to write down pH as well, somatotropin. That's growth hormone, that's its other name. Coming from the anterior pituitary, it stimulates growth. You have a lot of that early in your life, and it starts to wane later in life. It has periodicity through the diurnal rhythm. We often give that to elderly patients who can't put on muscle, who have trouble repairing themselves, that is with tissue. Somatotropin comes from the anterior pituitary and specifically affects liver, bone, but also write down muscle. Prolactin's another one, where it's going to induce mammary gland development. More mammary glands, more productivity in terms of cells within the mammary glands, and now you're going to get production. work done by those mammary cells. And indeed, they contract all the time in culture, secreting milk. The posterior, I don't want to spend too long on those. Oxytocin's involved in, anyone know? Labour. That parturition event. Remember we had that parturition event last week? The contraction, stimulation, but it's also involved in training your brain, imprinting the infant and the mother and anybody else who's close to oxytocin to say that that's someone I'm going to care for, I care about. So there's a lot of imprinting going on with oxytocin, not just this process of having mammary glands and smooth muscle relax in that case. Vasopressin, not going to spend time on that except that it alters how your kidneys work. Dialysis basically, altering salt content in your blood. Let's come back to our anterior pituitary. Some key ones in here. Forget this, luteinizing hormone, follicle stimulating hormone. Yes, it affects muscle accretion and bone mass and so on. The ones I want to focus on are this one. I introduced this last week, corticotropin or corticotrophin, aka ACTH, adrenocorticotrophic hormone. Last week I asked you to write down 1 to 39. It gets complicated because ACTH comes from this molecule. You're going to write it down, pro-opiomelanocortin. Pro-opiomelanocortin. It is a big, long peptide that's manufactured in the anterior pituitary and then fragmented. And we're going to see that later on today, fragmented to do different jobs. One of the fragments is ACTH. Pro-opiomelanocortin. We'll come back to it later on today, yeah? But just note POMC. ACTH 1 to 39 for the time being, because that's going to change as well. ACTH takes from my anterior pituitary we saw last week down to my adrenal. What does it do? Induces secretion of cortisol. The cortisol in your vesicles of the cortex of your adrenal are released. This is a chronic stress hormone. It's the same stress hormone we're going to see in obesity and diabetes, but also in anorexia. A disruption to this pathway can either send you more obese or more anorexic. And we're going to keep talking about those over the next few weeks. What is cortisol? Corticosterone. They are transcriptional regulators. They alter what you develop in terms of proteins, which is why I said the last 2 slides today we're just going to touch on. We'll touch on them next week again and the week after. The other one coming in here that we want to worry about a bit today is this one, thyrotropin or thyrotrophin, PEH. That molecule you're going to write down, producing TSH, thyroid stimulating hormone. That character regulates overall metabolism. You've heard about someone being kind of a little bit hyperactive, they've got a high metabolic rate. That's because of these hormones. What does that do? Well, that gets down to your thyroid. Where's your thyroid? Yeah, just here. It's a H-shaped gland up here. It's not the Y-shaped thymus, it's the thyroid. That thyroid produces a number of hormones we'll see today, T3, T4, the thyroxins. Very simple molecules. They get into cells and alter metabolic activity from a hormonal level. So there's our thyrotropin as TSH running down to form thyroxin, T3 and T4. What does that do? Alter metabolism, particularly in your liver, but also your muscle cells. Now remember, each of these, particularly these characters on the left, CH, adrenal, all those guys there are peptide-based hormones. They have cell surface receptors. And ACTH is a very labile hormone. It breaks down very quickly. We've already seen insulin, glucagon. Today we're going to see somatostatin. I kept hinting about that character as well. We'll see it again next week, regulating things like appetite. And our old friend over here, adrenaline, our acute stress response, also altering metabolism. This is the beginning of our hierarchy, but this is only the simple one from your anterior pituitary and a couple from your posterior pituitary. There are other hormones we have to start dealing with today. Can I move to the next one?
Two things, or just same thing with two names?
No, thyrotropin and thyrotrophin, PH or PIN, the same molecule, just whether the Americans or the English spelling. Okay. Can I move to the next one? Okay. Now I keep saying these are diurnal. I talked about how we're moving into the spring phase, which is mostly equivalent, essentially, light and dark cycles. I want you to look at that over here on this graph. We see here the dark phase shaded in grey, the light phase, obviously in the light phase either side, looking at a 24-hour clock time. What's that doing? And this is a normal clock time. We're not exposed to too many LEDs and the like. What you're seeing up here is melatonin. You're going to underline it because where's that come from? The pineal. What's it responding to? Day length. As the nights get longer, you're going to write down this peak of melatonin during the night phase gets longer as well. So melatonin is controlling that diurnal rhythm at a very high level based on exposure to light. How much light, how much dark. So you're going to write little arrows, one there and one there. If the dark phase increases, you stretch out that melatonin peak value through the night phase. When it decreases, you narrow it. That's controlling your diurnal rhythm. And if you're an animal in the wild, it's controlling your reproductive phases as well. When you're growing hair for winter, when you're shedding hair for summer, all of that's driven by melatonin, which is ultimately controlled by clock genes. Running down here, we're seeing, and I should just point out, this is in picograms per mil. Down here, we're seeing cortisol. There it is, nanograms per mil. What are you doing next week in the lab? Measuring someone's salivary cortisol. Now, cortisol has a binding protein. You're going to write down CBG, corticosteroid binding globulin. Its other name is transcortin. It carries this steroid around. It increases its half-life. You have to extract cortisol out to measure it, but in saliva you don't need to. You can measure directly like you're going to measure in the lab. But you can see cortisol, as we showed you last week, has a kind of an evening lull down here, down to this nadir, and then as it goes through the night phase, it starts to pick up. Right about sunrise, it's peaking and then it drops off. That's without a stress. That's just the normal pattern. Again, if I stretch out the dark, what happens to my cortisol? That dark phase is longer. If I narrow that dark phase, my cortisol phase is narrower. Does that make sense? So as I move the light and dark cycles, if I increase the dark or decrease it, I change this pattern overall by squeezing it up. What you should also know, and this is kind of critical as we go into summer, is that your body temperature drops off just by a degree or two during the dark phase. That's important for metabolism, but also brain function. It's good for your body to cool a little bit while you're sleeping. Allow your brain to recover. If any of you bury yourself under lots of doonas or blankets or have the heated up really high, the AC or the hot water blanket, whatever it is you use, you get too hot during the night, you feel kind of sweaty, uncomfortable, don't sleep well. That's because you haven't cooled your body enough, this degree or two, to allow recovery. So you should cool it a little bit. But you'll notice again, if we increase the dark phase, no matter what the temperature is, we'll then have a longer period of a slightly lower body temperature. Again, this is the normal phase without overlays. Now that's what we see for these patterns. Keep that in mind for your patient. Your patient number, you're measuring salivary cortisol, does it have an impact on the concentration, which might depend on when you sampled that patient, correct? Indeed it will. What we're seeing here is this diurnal rhythm as we run through this process, as I said, driven by clock genes, but we see it not only for that, but we also see it for glucose. What does ad lib stand for? I said you can go and eat ad lib. Ad libitum, eat as much as you want when you feel like it. Now, there are issues with that. If we let you eat as much as you want for as long as you want, what happens? We'll see the dark phase in grey, the light phase. Glucose tends to get a bit out of whack, doesn't it? It's not this kind of periodicity we've seen before. This is the general trend when you eat ad libitum. Snack, snack, another snack, another snack, rest, another snack, bit of TV, snack, another bit of playing on the phone, another snack. You constantly are taking in food, so it means your blood glucose is varying. If we restrict you to 6 meals a day, what happens? Looks fairly similar, doesn't it? Now let's have a look at corticosterone. Well, kind of looks like it should, shouldn't it? Peaks around the morning, that's fine. Light phase, dark phase. What about insulin? Why is insulin during ad lib 2 basic peaks when your blood glucose, shouldn't it pretty much match that? Sorry? Generally, yes. You're fasting more for that phase. Let's have a look down here for insulin on 6 meals a day. How many peaks have you got? 6, so they tend to match up when your ingestion, okay? So we're looking at the normal underlying rhythm versus what you ingest and what you do as an individual. What about glucagon? Well, it's not far behind, is it? 6 peaks, everything matches up, which is useful for us. If we look down here, we see adrenaline on ad lib. What happens during the light phase? Assuming no stress. You get into the dark phase, and that's not you on the phone playing some whiz-bang game trying to shoot someone up. It peaks for a while, doesn't it? It lasts for most of the night phase. What about growth hormone? That has periodicity too. Why is that? Because the anterior is getting stimulus to secrete at intervals all the time, which is what we see with growth hormone. What you want to write down, it's more prominent when you're young. As you get older, they tend to flatten out. Not such great peaks. They tend to flatten out as you age, and the overall level drops as well. We've just seen melatonin increase during the dark phase. Here's TSH. What do we say about TSH? It regulates metabolism. There it is, a busy day. I'm going to increase it. Then at night phase, that makes no sense. Shouldn't I be resting? But think about the night phase when you're recovering, particularly your brain. Have you seen those TV ads, those docos, where people go 8, 24, 72, 3 or 4 days without sleeping? What happens to your brain? Do you recall those docos? Sorry? You start going a little bit crazy, yeah, to some degree, but your ability to integrate knowledge, information and inputs really starts to drop off. It's almost like you're becoming a little bit underwhelmed or overwhelmed by alcohol, I should say. Your decision making is much more impaired as you go longer without sleep. So recovery for brain is critical. This gives you an idea of what's going on based on your diurnal rhythm, I have in italics, the day-light cycle, how much light and dark, but also what you do in terms of ingestion. Does this talk about a stress event like a metabolism lecture or an exam? No. It talks about this underlying rhythms based on what you're exposed to each day. Now, we're going to get ready for the day. Can I move to the next one? Now, this is relatively new. Just came out last year, towards the end of last year, as I was about to give the lecture. And I thought it's really important to recognise, because we're talking about clock genes, more and more are found in each tissue. And what are you seeing in this? What's a myocyte? Muscle. Have you ever noticed, now think about this, have you ever noticed, whatever your normal sleep pattern is, that you're lying in bed and, maybe you're not ignoring, or you are ignoring the alarm, but before you're about to get up, you feel kind of a little bit, not twitchy, but, a bit restless, time to go, bladder's full, where do I need to go, whatever, but you just feel like your muscles are just starting to move a little bit. You ever notice that? No? We're just completely zonked out, too much booze the night before. Too many cocktails. Notice it next time, because your body is getting ready for the day. Not because you've had a meal yet, but because your clock genes are telling your metabolism to get ready for inputs, to get ready for eating. That's what this slide's about. Let's walk through it. You've got clock genes hitting your pineal, registering light and dark, how much, what length of each of those. Those inputs tell you when you're going to eat. Let's go back one slide. When do you eat? Well, if you're on 6 meals a day or ad lib, I have a pattern. Write that down. I have a pattern. I'm priming, registering, and imprinting my genes, and that's a critical term for us. When you're first born, you are imprinted, just like birds are, animals are, of other species, to their parents or caregivers, whatever the case is, you imprint. But you want to jot down, you also imprint your genes, your gene expression. That's essentially epigenetics, where your expression levels are a function of what you're exposed to. So what do we got here? Well, this process alters your pattern through your brain. When you eat, how much activity you have, when it is. If you're doing a run first thing in the morning, like some of us, 5 o'clock, it's daylight savings, back to 6, but that's all right. Your pattern changes with what you do. You're imprinting, you're preparing yourself. If you've got a constant pattern, you are telling yourselves, now get ready for that process. I get through that process, I do work, whatever the case is. If I do work, I need energy. And that's critical, as we see here. What do you need? Glucose and fats. What does your muscle cell do? Well, yes, mate.
How does it work for someone who works in a night shift?
I get that question every year, and that's kind of really, really difficult. Depends on the shift. I worked shift when I was an undergrad, working for a big winery. The shift, if it's a rotating shift, can make life very, very difficult for you. Indeed, takes about 5 to 10 years off your life because of the rotating shift nature. Fireys, I know. I had one living across the road from me. He worked on 4-day shifts that would rotate from evening to night to morning and then 4 days off. It really screws up your cycle, I'm afraid. Whereas if you're constantly on night shift, then you can tend to adapt to it a little more. But there's different ways of running the shift work, I'm afraid.
But say you do night shift and then you go to metabolism workshop. How do you manage your sleep around that?
That's NMP, okay. That's right. No, that actually is very difficult, trying to get sleep, particularly during hot nights when you can't cool down, which is critical. You must cool down to let your brain and metabolism recover, ready for the next day. But if you can somehow get a pattern going so that you're training your metabolism for that cycle, does that kind of make sense? That's why the rotating shifts are the most damaging to metabolism and physiology, which is what you're doing in this green muscle. Hopefully there's not too many green muscles. What you're doing before you get busy, you're actually getting cells ready for the input of ingestion. So even though I've talked about glucose transporters being put on the surface after a meal, you're actually priming the metabolism, getting the glucose transporters ready close to the cell surface, so the moment you have breakfast, everything's ready to roll. Does that kind of make sense? So your tissue is preparing for activity, not just the insulin stimulation of suck up glucose and do work. And also think about that. What's the liver supposed to do during the night phase? So you're in a fasting state, aren't you? So I talk about all semester, fed, fasting, and the stress state. So if you're in a fasting state, you want to get ready for input of food, but also activity as well. Does that make sense? Okay. That's critical for us because what you're doing is altering. You're going to write down imprinting your genes for daily activity, for your daily cycles of eating. And again, what can help you on these shift works is to have meals at certain times and also be exposed to light, because particularly if you regulate the light that you're exposed to will help you sleep, so you artificially fool your pineal. Particularly blue light is a problem, like all those LEDs on your computers and your phones are the worst ones for screwing up your melatonin. That's why parents in the know tell you get off your damn phone and computers so you can get a normal sleep pattern, particularly during puberty when you're most vulnerable. That's why you're in this room. A lot of your colleagues during puberty didn't focus as much. They couldn't focus on the work. Their brain was being rewired. Bam, they're off doing something else, and you happen to be in this room.
Does supplementing metabolism help melatonin help at all?
People do melatonin pills often if they're doing flights around the world trying to catch up. It helps to some degree, but the problem is you've gone from, let's say, imagine we're here in Sydney and we head off to LA, different sleep cycle, different light-dark cycle. I've done it a number of times. I know it takes me 2 or 3 days to adapt over there. I come back here, it's only a day or two. But if I'm over there longer, I imprint longer, everything takes longer to come back to this light-dark cycle. Melatonin doesn't help adapt metabolism as much, it just makes you get used to the light-dark, if that makes sense. So you can be sleeping, but not really sleeping, recovering, if that makes sense. Good questions. Okay. Our GLUT4 transporter. Go back to what we saw, GLUT1, GLUT2. What's that one? Involved in muscle particularly. Taking up glucose ready for activity, which is kind of critical for us. These are genes telling us that this needs to be done, ready for the day's activity. Now, if you were back in the dark ages, running around with sticks and spears, you had a fairly reasonable cycle, didn't you? No lights to switch on, no computers. When the sun went down, that was basically it. You weren't exposed to UV, the wavelengths that cause your pineal to change. It's just a fireside chat. Can I move to the next one? Okay. Now you've got a bit of summarising to do to some degree. I know it's a bit wordy. But that depends on the type of muscle you have. We're just talking about marathons. No, that's different to lifting weights, to doing sprints, for example, which means the type of muscle, which is actually tested in dogs and horses that run races, and humans now to see what kind of muscle they have. Muscle forms means that you can actually get them ready, which this didn't roll all the way down. The types of muscles you have, you probably know that yourself. Oh, I couldn't run a sprint to save my life. But you can also train muscle on how many mitochondria, what genes, what activity, what proteins are involved. Slow twitch muscle, fed by a lot of blood vessels, they have a lot of mitochondria, produce energy by slow, steady, ETC, what's that stand for? Electron transport chain and ATP synthesis. Fast twitch muscle, they tend to be less coloured, less red. Why? Just like we saw with fat cells, more mitochondria, they tend to be darker with all the cytochromes. Fewer mitochondria, lower oxygen, they use ATP faster and they fatigue faster. So if you were an endurance runner running marathons, which muscle type do you think would you predominate mostly? Faster or slower? So endurance training will do what for you, do you think? Increase it. You're stimulating protein accretion, more mitochondria. That's why you should go to the gym or do whatever exercise you need to do to keep the working process in cells. It will increase your lifespan. You use glucose, fatty acids and ketone bodies when? When the ketone bodies? Once you start to push beyond what glucose is available. Your resting muscles, including your heart, is mostly using fats, less glucose. The brain's using that stuff that I keep talking about. Can I get to the next one? No? So if you were a sprinter by design or genetic background, breeding or otherwise, should you try and become a marathon runner? You could try. It's always worth a try, but it's going to take a lot longer, isn't it? You're never going to reach that. I was just telling one of your colleagues, I was running with some distance runners from Ethiopia when I was in California, and they can run. Man, they can run. Can I move to the next one? Ready to go now? All right, they can run because they're making ATP. Now, remember I said a few times that you make ATP and you send it into the cell cytoplasm and then you do work with it? But I said if I get you to stand up here and then run around this campus, hopefully not run into anyone because not many people are around, it's going to take you a few metres, 10, 15, maybe 20, 30 metres before you start puffing. Well, that's incoming oxygen to make more ATP. How did you last those 20 or 30 metres without kind of running out of ATP? Well, what you have is kind of this buffer reserve. You know, have you ever driven an old Volkswagen, the one with a little reserve tank on it? Well, your muscles have this reserve. They've got this reserve of creatine, which has got phosphate on it, but it's not ATP. And what we do is we take the creatine phosphate and make it so it's available as ATP in the cytoplasm. What does that do? It saves time. There's my energy reserve sitting as creatine phosphate. So as soon as I have to start running away from some tiger or lion or some metabolism academic, I can get quickly before I have to start breathing more and making more ATP. Does that make sense? So I've got this buffer ready for quick work, which is what we're seeing here. If you stress your muscles, which means get up, run now, do it quickly, but don't do it. Okay? What are you doing? Well, you need to get work running quickly, so it's bursts of what is termed heavy activity. You're going to change quickly, trying to use whatever you've got. What might happen? You'll form lactate. Didn't we talk about lactate cycle earlier in the semester? Yeah. Using those carbons to make something else like glucose. I run through that process. I produce CO2 because I'm an electron transport chain. But these bursts of heavy activity also grab phosphocreatine right down in the muscle, particularly in the muscle. This phosphocreatine gives up the phosphate to help make ATP in the cytoplasm of the cell. We don't have to run through the mitochondria anymore. It's already out there. We've got it stored ready. And that helps with the process. Now, that doesn't mean you don't need to replenish that and make more ATP. That's why after 10, 20 metres, you start breathing heavy. What are you doing? Inputting more oxygen, trying to make more ATP as quickly as possible, importing more glucose or fats, whatever the case is, or ketone bodies, to catch up as quickly as possible. But that phosphocreatine is the reserve ready for you to get away in a hurry. Think about one thing: flight-fright response. Acute stress, what do? With bursts of heavy activity, fight quickly to overcome the perpetrator or run away quickly. The first 20, 30 metres enough to get away. That takes too long to go through what? Glycolysis, the citric acid cycle, and ETC, make ATP, then send it out. You'll be dead by then, wouldn't you? That's why your muscle must have this phosphocreatine reserve. So you don't wait for that to catch up. Yes, it will take time. So if you're a sprinter or a weightlifter, that's been too long, isn't it? How long do they take to get 100 metres now? Less than 9 seconds? Yep. That's about the time it takes to get all of your energy back on track. So they're all built up, ready to go. Distance runner, what do you run? 2 hours or whatever? Triathlons, even more. So you can't rely on that phosphocreatine. You've got to rely on the mitochondria getting energy in and getting things working. That's why endurance training encourages more of that. What have we got with our phosphocreatine? Well, I popped over here your old mate. We start there with glucose, and we went down to glucose 6-phosphate. We've trapped it with hexokinase. We ran down there, down to pyruvate, acetyl-CoA, the citric acid cycle, alpha-ketoglutarate, succinyl-CoA, OAA, lots of protons, respiratory chain. You're going to write down, I've seen that before, but that takes time. Too long for me to survive an attack. Flight-fright response. I must get away or I must fight. That won't save me. That's why you have phosphocreatine in your muscle cells, particularly for that process. Who stimulates that process? Adrenaline. Brain tells you, dump it out quickly. That one of those short answer questions will be fed, fasted, and stress state. What's the stress state? The flight-fright response. Can I move to the next one? Yes, mate.
So you said that once the buffer is gone, then is the process increased, or was it not happening in the first place?
Can you just hold off for that for the next slide? I'm about to take you through that, but it's a really important point. That's why it's on the next slide. Can I get to the next slide? Is that a yes? Okay. Well, it's the slide after, sorry. What's creatine? Here it is here. Excuse us. What's creatine? A couple of nitrogens, a couple of carbons, and a C-bell bond O. What do? You put a phosphate on it. There it is there. I can assure you that won't be one of the energy carriers in the final short answer question, the multiple choice, I should say. What do you do with it? That's your buffer. As soon as you need it, what have you got? You form creatinine. You're going to write down, you can measure creatinine in blood and plasma, particularly, and urine, sometimes in saliva. It will tell the practitioner, the GP or otherwise, whether you're using a lot of this kind of molecule as our creatine. Who does the work? A kinase, no surprise. And if you are producing lots of that, it tells us what your muscle has been through, which is what I'm going to get to, I promised, in the next slide. Starts off life here as glycine. You need to know this chemical pathway. Glycine with arginine, I've seen that many times. What was arginine important for? Who particularly? Who? Young child, adolescent, in growth phase, indeed. What do? Run through this transferase. You form this guanidinoacetate, incoming methionine. You do another step. What have you got? Creatine. You put a phosphate on. What have you got? Phosphocreatine. There's your energy buffer. What do? You hand off that phosphate to somebody else, which is what we have over in this simple schematic. We've spent all semester making ATP. Look at the schematic. Making ATP, sending it out of the mitochondria. But just note here, in the intermembrane space of your muscle cells particularly, you have this thing, a mitochondrial kinase, a mitochondrial creatine kinase. What did you do in the intermembrane space? Phosphorylate creatine. That's it going on here. Out comes your phosphocreatine. There it is. Look at my laser. There's my phosphocreatine. What do you do with it? Well, it hangs out there until you have to do bursts of activity, muscle work. What do you do with that phosphocreatine? Well, you're going to keep cycling that phosphate around, ready to make ATP. ATP is what's required to do the work. Let me show you again. That finger contraction. Well, I should do it here, shouldn't I? In advanced cell, we talk about this. You have actin fibres, lots of them. You have myosin head groups. They work on each other. So as these two contract, that contracts like that. Drags in the muscle fibre, your ligaments, to drag the muscles together. That's what happens there. What you've got to do is move this protein, troponin, tropomyosin, out of the way for the head group to grab. In this case, calcium moves it out of the way. This motion, it's like a golf club head grabbing fibres, is driven by ATP. It actually doesn't induce the contraction, it induces the relaxation, which is kind of odd. But that muscle contraction, that contraction is ATP driven, controlled by calcium. Keep that in mind. So now we've got this turnover. So we have a mitochondrial creatine kinase, and the CCK is not cholecystokinin, it's cytoplasmic creatine kinase. So I can keep cycling through, putting phosphates on creatine. What is this muscle doing essentially? It's saying whatever phosphates I've got, because I might need to do a burst of activity, rather than just relating to ATP and relying on it, I'm going to create this buffer of phosphocreatine, so the moment I have to work quickly, I've got my buffer to do that. Does that kind of make sense? So my phosphocreatine is my buffer for bursts of activity, which is what we're seeing here. So any free ATP running around in the cytoplasm, your muscle cell puts it back onto creatine to make phosphocreatine. Can I move to the next one? Okay. Now this is answering your question. I need you to not write, I need you to look down at the bottom, and I need you to look at this as a three-dimensional graph. Don't write anything, because this graph is kind of critical. A few exam questions on it. What you're looking along here is measuring inorganic phosphate, measuring phosphocreatine, measuring the amount of ATP in a muscle cell. Don't write, just think about it. So what I'm doing, I'm measuring ATP, phosphocreatine. What about these guys? Inorganic phosphate. Well, if I've got inorganic phosphate, it means I don't have ATP anymore, doesn't it? I've done some work with it. If I look along here, these chemical shifts, let's ignore them for the time being. We're looking at an amount, that's what the peak values are. What I need you to do with your pen now is you see that red exercise. I want you to draw a line through there, essentially through that dip, through here, right through there, all the way through. And then another line on the other side, in between exercise and recovery, through there, just through that dip, and then the other side of that peak of phosphate. What's happening there? We are measuring the amount of inorganic phosphate. If I see lots of inorganic phosphate, it means some ATP has been converted. So I've used up the phosphate as ATP. What does it tell me about the ATP peaks you see along here, 3 of them? Did they change when you exercised? No. Keep that in mind. They didn't change. What did change during exercise? The amount of phosphocreatine. Now I've got phosphocreatine, I exercise for a while, what happens? I've just got creatine now. The phosphate now is being used to phosphorylate somebody, I have inorganic phosphate. Why is that? Tell me why that is. Try again.
So, your body's doing the exercise and it's using that phosphocreatine.
Yep, that's good. Yep.
The off-product of it being used.
Good. So that tells you that's how much buffer of energy I've got available in my muscle cell. Okay, I can work that out. But why do these 3 don't change during exercise? What does that tell me about the cell's metabolism? I'll give you a hint. To produce ATP. So how much ATP am I producing during my resting phase, during my exercise phase, and during my resting phase? Does it change? No, because the amount of machinery, enzymes, the number of mitochondria you've got, that didn't change in that time frame. We're only talking a few minutes here. We're not talking about days and weeks. So what does that tell me about my activity to make ATP with the number of mitochondria and the machinery? Did it change? No. So even when I'm exercising, so that's why I asked you to draw a line through there, running around the campus, do I increase the amount of ATP? No, that's relatively constant. So if I run around the campus and I haven't been training, that's as fast as I can go or I'm going to stop, right? To be able to get faster and go longer, what do I need to do? Train, get more machinery, more enzymes, more mitochondria. That's what training does, particularly endurance training. It's encouraging you to lay down more machinery, more mitochondria to do work. So if I looked at an athlete who was a really good distance runner, would you expect these AT peaks to be a little bit higher and maybe the phosphocreatine to be higher? Yes, you would. If I looked at somebody who's just playing on their phone all day, who would that be? Would their ATPs be relatively low? Yep. So that means even when you're inhaling a lot of oxygen, you're not actually making any more ATP, are you? Any more than you're capable of making. Does that kind of make sense? And that's why you'll stop at some point. Now this happens over time. That's why I have this other graph up here to show you what happens over the first few seconds and minutes of that burst of activity. Do you want to finish writing down the bits you need to, and then I'll talk about the next bit? Yes, ma'am. Yeah, I knew that question was coming.
Because if you're stopping yourself from actually making more metabolism, wouldn't you? No? I don't know.
Yeah, keep going. You're on the right track, yeah.
In the long run, it would.
So here I am. I'm a gym junkie, which doesn't look like it, but never mind.
Because by taking the creatine, you have these extra supplies, so your body isn't changing its metabolism to make more machinery.
Yeah, so let's say I'm not a gym junkie. I just take creatine supplements for various reasons, and I just play on my phone all day. So what would your metabolism, the cell machinery, the amount of mitochondria, does that change? No.
No?
And indeed, you just pass out most of the creatine. You don't actually use it, which is a bit pointless. But if I take those supplements, you're not actually changing your machinery. That's why exercise is critical, because that's increasing the amount of enzymes, proteins, mitochondria to do the work. Then you might find phosphocreatine gives you a little bit of a boost, but even then, as I said, most of it's passed out. Doesn't do much. It's got to be in the cell. Where has it got to be? It's got to be, let's go back a slide. It's got to be in your mitochondria that it's actually phosphorylated there. Kind of make sense? Okay. And to get back to your question, if I'm having trouble making it, because there's a defect in this pathway, it might be useful, and some diseases are the case. But in this case, if I've got lots of arginine, well, that's not usually a problem for adults, is it? So it's not going to be a big issue. So it tells you now that exercise is critical to up-regulate all of these particular proteins and components. Can we talk about this graph now? What happens in the first few seconds? You use up your ATP. There it is, bam. What do you do next? You grab your creatine phosphate. Notice that's seconds before you hit the minutes. So here we go, up, out for a run. Seconds gets me to where, do you think? Edge of building 21? Yeah. Is that where you start puffing? Pretty much. Now I'm going, I've just used up all my creatine phosphate, so what's my activity level after I get past 21? Well, I'd probably slow down a lot. But in the meantime, I'm going to use a lot of anaerobic activity until I can inhale enough oxygen to run through, let's go back here to your mate, run through all of this pathway and here to use that oxygen to make more ATP. Make sense? But the rate of activity, the amount you make, doesn't really change. Comfortable with this one? So we're all going to be down the gym now, are we? Or start running marathons? You won't catch me in a gym, I'm afraid. Okay, get to the next one. All right. Sorry?
Membership with our enrollment.
I have plenty of work to do at home. I don't need any gyms. Let's start talking about how you use hormones to regulate activity. And I've kept talking to you all semester about how your gut controls your brain, your fat cells control your brain, and you think your brain's controlling everything else, but it's not. We're dealing with a number of hormones here in yellow boxes. I'm going to step you through this kind of slowly. A number of concepts today. First of all, we start off with clock genes and a hormonal hierarchy from your brain, your pituitary. Those clock genes then say, this is my pattern, my activity level. That's what I do at the brain level down. That's why it's called a hierarchy. My muscle's ready to go, depending on whether I'm just slothing around or whether I'm exercising. That depends on what experience I've had, whether I'm trained for it or not. But here, your gut, your stomach, last part of your stomach and duodenum, just past the gallbladder and down in your colon and beyond, you have a few hormones. And these aren't the only ones I need to alert you to. These are a few characters. Just want you to look at the schematic of your gut for a start. You're going to write down a few things, hopefully for you to remember. Your stomach produces grumbling ghrelin. Quite literally, you feel your stomach grumbling, all the acid turning over, bubbling away. You feel hunger, that's ghrelin. Secreted by the cells in your stomach. Does the stomach absorb anything? No, it's a big muscular gland, only vitamin B12, drugs and a few toxins. But it secretes grumbling ghrelin. It's going to be an exam question on that. That tells your brain you're hungry. Tells your brain your stomach is empty, you need something to eat. The lower part of your intestine, just past the duodenum, produces CCK. What's that? I asked you to write it down earlier in the semester. Cholecystokinin. What does that character do? Stimulates gut activity and suppresses appetite. What does that mean? Somatostatin says, well, you're kind of full. Things are moving through. What do you want more food for? You get down here to this one. GIP, the lower part of the duodenum. What's GIP? Glucose-dependent insulin inotropic peptide. Mouthful. Just think of GIP. The next one down in your lower colon is GLP, glucagon-like peptide. Oh, you gotta love biochemists and endocrinologists. Worst acronyms ever. What do those do? These are incretin, not cretin, incretin. We're going to come back to that. Peptides, that is amino acid-based hormones that induce insulin and inhibit glucagon secretion. So if these hormones are coming from your gut, GIP and GLP, they're inducing insulin, what does that mean? What's it going to do to your metabolism? What's it going to do to your glucose transporters? What's it going to do to your liver? Let's bring down the buffet. Here it is at the front, big buffet. Can't touch it yet, but your stomach's seen it. What's it doing? It might be time to get something to eat. Okay, now I rush down to the buffet, trough it in. It's going to take a while before it gets in here, processes there, gets out there, absorbed in there, gets to your beta cells to do what? Make insulin. But what's your gut telling your beta cells already before you even get to the food and before it gets too far down your gut? It's telling the insulin beta cells to get ready for the incoming food. Does that make sense? Not just here it is now, let's respond to it. Here it comes, let's get ready for it. Best way to think about it. Which is what you're seeing here. Your gut is telling your beta cells, other islet cells, to get ready for incoming food. Prime the system. Make sure your insulin's in vesicles, lots of it, because there's a big smorgasbord coming in, getting ready for the intake. Let's go back to one of the first slides. I won't flick you back all there, but think about your diurnal rhythm. What happens just after you wake up, apart from going to the toilet? Head to the kitchen, raid the fridge. Your stomach might be rumbling at that point if you haven't anything to eat the night before. Later in the day, what are you doing? Just before you have lunch, before you have dinner, your stomach's rumbling, telling you get ready. Food goes in, get ready. Insulin, get ready. Which is what you're seeing through all of this. But like we saw in the first half of the semester, we saw feedback inhibition of products, didn't we? If those cars built up in the car yard, they feed back to other enzymes to say slow down or shut down. Well, you're seeing the same thing with hormones. Feedback inhibition. If lots of these hormones are running around your blood circulation, that's what these arrows are over here. Keep in mind these are hormones in your blood mostly, some in lymphatics. in your blood feeding back. Let's have a look at one for a start. GIP, incoming food. What does it do to our alpha cells? What's the red cross mean? It says food incoming, glucagon turn off or slow down. GLP from my intestine, incoming food. What does it do to my beta cells? Food incoming, get the insulin ready. So I'm ready to go. So you're priming a system, and your diurnal rhythm's priming it too. Your clock genes are priming you ready for that activity. What about my grumbling ghrelin? There it is. Oh God, I'm hungry. Now, the more I think about this smorgasbord. Grumbling ghrelin gets back to your brain, tells particularly your hypothalamus, get ready. Your brain is focused. Now, what you also want to jot down, often the hungrier you get, the more acute your brain starts to work. What does that mean? Well, they know, a number of researchers have done work on this, that the more you fast, the more your brain becomes active to problem solve. Because hunger means you need to get food in to survive. If you don't survive, NMP. So grumbling ghrelin tells my hypothalamus, get busy, work forward. What does glucose do? Get busy, work forward. What does it do? It tells your hypothalamus, time to eat. Go and grab something. Does that kind of make sense? You're priming everything all the time on the circadian rhythm based on day length, based on what you eat, based on what you do. A couple other ones. Our gamma cells, I mentioned before, somatostatin has an effect as well. We're going to see what it does in a moment. You're also seeing come from your intestine somatostatin. My colleagues in CSIRO used to immunise against somatostatin. It's like making antibodies to alter the half-life of somatostatin so that you get increased rates. You inhibit the process of appetite suppression. All of these are driven by kinases. There's a surprise. All semester we've seen kinases, particularly these guys, the AMP kinase. What do? They cycle around. What your blood glucose is, high or low, we're going to monitor that process. If my blood glucose is low, what's my brain getting a signal to do? My blood glucose is low. What's it telling it to do? Go and get something to eat. Totally. Think about that during the exams when you talk about the fed, fasted and stressed state. We're ready for the next one. So yes? Okay. I keep saying you have to overlay this with diurnal activity, diurnal rhythm, how much light and dark. And this diurnal glucose, ghrelin and insulin I'm going to focus on for a little bit. Ghrelin is a short-term underlying orexigenic. We're going to see that term a bit as we go through today. Peptide, which means amino acid based, secreted by the stomach. Look over to the right. Put your pens down. If you look all the way down the bottom, you see the time of day, 8:30 a.m, and that's when most of you are crawling out of bed, just before breakfast. Noon, 5 p.m, the dinner time, and evening, a little snack, and then off we go. So there's your diurnal rhythm. Where would you put the dark phase roughly, do you think? Let's make it equivalent. Around here, yeah, around 6 till around 6 a.m, for example. Let's look back up at the top. What's happening here? The dashed line, 8:30, 12 p.m, 5 p.m, 8:30. You're going to write down that is food intake. Breakfast, lunch, dinner, snack. I don't like the word snack, but never mind. That's roughly 4 hours, isn't it? I kept saying that before, unless you snack in between. What happens about mid-morning for me? Early morning for you? Grumbling, go get something to eat. So I have a peak of ghrelin just before I go and grab something, telling my brain, go and get something to eat. Same thing when I get around lunchtime. Getting hungry, now I go and get something to eat. Same thing dinner time. There's a few snacks in the afternoon. So the grumbling ghrelin is linked to not only diurnal rhythm, but what you do during the day, when you eat a meal. If you look down below, you see blood glucose. Well, I've had a meal here. There's my glucose go up. The shaded area indicates some variation. Standard deviation, standard error, doesn't matter. There's my meal, another meal, blood glucose goes up, a little snack in the evening. It's pretty straightforward, isn't it? Now let's have a look at the bottom one, insulin. Matches up as well, but note, ghrelin comes first, then glucose, then insulin. Does that make sense? That's the sequence of events that you're ingesting because your stomach told you you were hungry.
Why does it continue to stay high after snack? Like, why doesn't it go down?
After where, sorry?
If you look at the graph where it says snack,
Yeah.
snack continues to keep going up.
Yeah.
Why is that?
What do you think's happening during the dark phase? And let's assume now that around here, around 8:30, maybe 6, what are you going into? Sleep time. So you're not necessarily having enough material in your stomach. You're switching to what during sleep time? Sorry? Fasting state, yep. And what kind of activity is going on in your liver at that point? Or transitioning to, I should say. Glycolysis or gluconeogenesis? Gluconeogenesis. So to answer your question, hopefully by then you're having nice sweet dreams. Hopefully by then your stomach's pretty much emptied out, or at least transitioning through. Hopefully by then, and through the middle of the night, you're transitioning from glycolysis to gluconeogenesis. So you don't need a full stomach. Your brain's switched off. You're relying on gluconeogenic pathways to keep everything ticking over. But didn't I say at the beginning of today, during the dark phase, your body temperature drops off as well? So your activity tends to drop as well. Does that make sense? Yeah. To refresh and repair and overcome all the issues you've dealt with during the day. Okay. Does that answer your question? Sorry.
I'm
No?
a little bit confused at why the ghrelin though doesn't go down. So I understand that you're changing to the other one, but why after eating it doesn't it go down?
Doesn't it go down because it's a snack?
It's not enough.
It's never enough, is it? And actually, look, it kind of leads into the next few slides we're talking about. Your stomach expands. The stimulation of that expansion sets off a whole set of hormones that says now your stomach is full. What you need to think about, have you heard of glycemic index by now, I hope? Yeah. The Romans used that. They didn't know about it, but they were using that to have glycemic index foods that allow their armies to march longer, work harder, without having to refuel. What that's doing is I can fill up my stomach, but if I have a lot of McDonald's, Hungry Jacks, what happens to that stuff? It kind of turns to mush and it passes through very quickly. I expand my stomach. You've seen those TV shows where some character's eating hot dogs or hamburgers, sausages, stuffing it in like some TARDIS. Yeah, your stomach expands. And as it does, it sends out signals to say it's full. If you ignore those full signals, then you start to have issues because your stomach keeps on expanding. I'm going to talk about the 5-2 diet over the next few weeks as well. Keeps on expanding and it doesn't say to your brain, well, it's full, why are you still eating? And the brain says, well, because I've got an addiction to food we're going to see at the beginning of today. During the snack, you didn't fill it up enough. Just a snack. Keep you going. Now, that's not enough, and that's why it stayed high, because you haven't filled up the stomach to get a feedback to say, now the stomach is full. But at that stage, the brain says, I don't care. I need to have some sleep and repair and recover from the day's activity. Does that explain a bit better? Okay. And it may be time for a break. Yes. And go and have a snack. That's right. Especially the free ice creams that are given away.
Question about the
One second. Shift work.
Yeah, my dog woke me up at 4 a.m.
Oh, no.
I know, what a pest.
Are you still a puppy, though? Or
Yeah,
kind
10
of?
months.
Yeah.
I love that guy.
Did he have to go to the toilet or something, or was he just being
No,
difficult?
he was just like, he wanted to get under the blanket, so he was like scratching. He did my team leader speech this morning.
Cool.
Again, it was fun.
Cool.
He didn't ask me anything.
Oh, that's right. So Parramatta, isn't it? Yeah. Alex. It was Alex, wasn't it? Yeah. Because he was covering for Hamid the other day. So.
Oh, yeah, we had him in once, I think.
Yeah, we did.
Yeah.
One, I think one, two.
How are you?
I'm good. How about you? Apart from no sleep.
Pretty good.
Huh?
Could go for a nap, but I have this annoying pharmacology report. Let me tell you, not every lecturer is as good as you.
Right. No.
So it's painful. It's genuinely, this report has been so drawn out. Like the first tutorial of the semester was on this report. There's been 2 drafts that need to be submitted for it. That weren't given you. Literally, you had to submit it and turn it in. And they were like, oh yeah, we'll give individual feedback. No feedback was given. And they weren't marked.
Okay. I appreciate the offer. Okay. I said I'd walk you through this stuff kind of slowly. I said it was a bit conceptual. There's a number of concepts today, thinking about clock genes, hierarchy. We had receptors last week, signal transduction last week. All of these are underlying everything I'm talking about. Every one of these receptors, every one of these hormones has a receptor, either a cell surface or an intracellular. I showed you this just a moment ago with some bits missing, and this diagram as well with some bits missing to step you through it slowly. We've already talked about GIP, GLP, ghrelin, its relationship to those characters we've seen all semester, particularly glucose, insulin, glucagon. I've hinted at somatostatin, and today I just mentioned at the beginning that you can change your overall metabolic rate from molecules coming from your thyroid. Those molecules start life as thyroglobulin, which I'm not going to go through all the biochemistry and chemical structures, but that polypeptide is broken down into thyroglobulin, and that thyroglobulin has a tyrosine, and it's not phosphorylated. It's actually iodinated. If you eat lots of seaweed, lots of iodine in seaweed, you'll have lots of T3 and T4. If you work in an isotope lab, like I did for many years, and you use iodine 125, which is a gamma-emitting isotope, you have to monitor your thyroid. People working on radiation have to look at it all the time because your thyroid collects iodine. which is a useful thing because with that iodine, you put an iodine on your tyrosine residue, you then cleave off the peptide. What have you got? Thyroxine, T4, or triiodothreonine, T3. I'm giving you T3 down here, a couple of ring structures. See those little eyes there? 1, 2, 3. This is a metabolic regulator. This hormone starts life in your thyroid, released into your circulation. It goes into cells. It doesn't have a cell surface receptor. Into cells and stimulates overall metabolic activity in cells. Particularly people who are a bit hyperactive, you'll see them. They don't wear as many clothes, they're not rugged up, keeping warm, they're kind of bouncing around, a bit type A in some cases. They're busy with a high metabolic rate. Kids are like that too, aren't they? Too much raspberry cordial, we're bouncing off walls all over the place. And then they crash. This T3 molecule we're seeing here, what does it do? Well, it comes from your thyroid via the pituitary, TSH, all the way down here, released, changes metabolism of all of your cells, and it goes back to the hypothalamus and says to the hypothalamus, you're already hyper. Now you start slowing down because you've got feedback, note the red cross, inhibition. Too much T3 or T4, you feed back to the brain to say, don't keep making as much. It's feedback inhibition. Make sure you've got that term. The other thing we have to worry about on this slide is this other character here that comes out with GLP in the lower part of your gut, near the ileum. And I know there's lots of crazy acronyms, but this one's called PYY. There's a number of hormones called PY, protein Y, peptide Y. This one's called pancreatic peptide Y. You'll see the YY, and 3 to 36 means it's amino acid number 3 to 36. So it's only 33 amino acids long. What does it do? Well, when your ileum is full, what is it telling your brain? I'm full. Why are you still eating? It suppresses appetite as well. But you can override these issues, which is what I'm going to start talking about now. If your brain overrides them, you keep eating, expanding your stomach when you shouldn't. Now, before I get to the next few slides, I need you to think about a few things as we go. We've talked in the first half of the semester, metabolism of cells, turnover, feedback, all that stuff, just basic grunt biochemistry. Integration's about the hormonal control. How hormones control what cells do based on what you do. But what we have to keep in mind is what you eat, the nature of the foods you eat, high glucose, for example, high carbohydrate or high fat. If you're on a high protein diet with nothing else, what does that mean? I've got to rip off nitrogens and make them into glucose if I was a carnivore. But as you go, you need to think about receptor number, activity of cells, but also what your brain perceives as what it wants. Because food is an addiction. The sensations are the same sensations as gambling, as drugs, as in the ones that are neither naughty or illegal. Those are the same sensations as your brain, which is what I'm going to get to at the beginning of today, which builds to all these issues like obesity, diabetes. And that's what we have to think about in the context of not just metabolism in the cell, integration of the hormones, it's what your brain is perceiving what you want to do as well. How big your stomach is stretched, how much, what signals it gets, and how many fat cells, how much fat you've got to feed back to what your brain perceives. And whether you listen to those signals, think about the receptors, the response, the integration in your brain, or whether you ignore those signals because there's an addictive behaviour. The hormones controlling addiction are the same. So keep that in mind as we go through the next few slides. And can I get to the next slide? Yep. And this process, you can see here, we've got more feedback, inhibition. What have we got? Insulin coming in on the right-hand side. Last week, I specifically showed you the insulin receptor, didn't I? Kind of like this muscly guy, inside the cell, these two arms, and they phosphorylate each other, like I showed you the dimer. Well, that's for a signal transduction for insulin. What you need to note now is the crosstalk between that and other receptors. I showed you that process earlier on with the insulin receptor. Well, that crosstalk starts to happen down here. What have we got here? This character called leptin. Leptin is elevated in many cases of obesity. And administration to most obese people does not restore normal body mass. Why should that be the case? Well, leptin's telling your brain you are kind of full. Here it is. You see the schematic. Here's your fat cells. Lots of fat in your fat cells. Leptin's produced. So there's a hormone produced by fat cells. Leptin's produced by fat cells. It goes back to your brain and it feeds back to your brain. Your fat cells are full while you're taking in more food. You don't need to. You don't need to store any more. You are at capacity. But your fat cells also produce this thing called adiponectin. Adiponectin, when you have low fat, there's my fat cell, but it's kind of shrunk down, not as much fat in it. It feeds back to your brain. What does it do to your brain, the green triangle? If my fat cells have shrunk, there's not much fat into it, what would it tell your brain to do, do you think? Go and eat some more. That's what it's doing. When these are out of balance, that's when we have obesity, diabetes, and all those things coming into play. But please circle that. That's a fat cell producing two hormones. One says you're full, slow down eating. One says you're getting close to empty, speed up the eating. Fat cells produce hormones. Gut cells produce hormones. And what happens here in a cell? What we've got, this plasma membrane, is our insulin receptor. There's our muscular character, click, 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 and then all of a sudden we activate a cascade. And that same cell has receptors for leptin, as it has for most of the hormones in your body. That's why I talked about the room where you're sitting in, playing on your computer. The kitchen says dinner's ready. You're integrating signals. Well, your cell's doing the same thing. Except now specifically, you're seeing this crosstalk between the leptin receptor. You see that Jack there? That's not Jack, the PhD student I had. That's a Janus-associated kinase, not Jonas, Janus, associated kinase. Immune cells use JAK kinases a lot. Leptin receptors use JAK kinase as well. What do? They get involved with this intermediate receptor molecule, which is heavily phosphorylated. What does that do? Activates PI3 kinase, we've seen all semester. What does that do? Stimulates cellular processes at a metabolic level. Insulin says, blood glucose is high, you don't need to eat more, run this pathway. Leptin says what? What is leptin doing? Says your fat cells are full. The two are telling your cell, you don't need to take any more in. The two are telling your cell, inhibit food intake. You are full. You are capacity. Your tank is full. You don't need to fill up the tank anymore. But what leptin, not the tea. What leptin does, and I had a leptin before I came down, leptin makes the liver and muscle more sensitive to insulin through what we call this common second messenger, this IRS protein, to stimulate the process. Now what happens is, in your brain, or some people's brain, we get what is termed leptin resistance. Now let's think about that for a moment. Leptin has a receptor. How many receptors on the surface? How much signal transduction? If I put a lot of receptors on the surface for leptin, what happens? My cell is more responsive, right? But if I take them off the cell surface, what happens? My cell is less responsive. If I change the signaling cascade, it doesn't work as well, what happens? Cell doesn't respond as much, which is leading me to next week and the week after when we see insulin. Insulin resistance, insulin sensitivity, changes with receptor number and signaling, as we'll see next week particularly. Whoops, this is building that process. The other thing that comes in is this appetite-stimulating protein called neuropeptide Y. I know they keep coming at you, don't you? I've just got those covered. Now there's another one. Neuropeptide Y, small amino acid sequence. It inhibits release of appetite-stimulating neuropeptide Y. That's our character here. It's an orexigenic neuron that does that, our leptin-producing one. What insulin also does is it's involved in this guy, alpha MSH, melanocyte stimulating hormone. We're going to see that in the next slide. You're just going to put next to that, that started life in POMC, a big long polypeptide that was cut up into fragments related to obesity and anorexia, one or the other. alpha melanocyte stimulating hormone. And melanocytes do what that you know about? What do they do? Where do you see melanin? In your skin. And as the sun comes up, you get more beating sun on. What do? You move melanin vesicles to the surface of the cell so that it takes up UV. Doesn't hurt so much in terms of your UV damage to your cells, hopefully. Can I move to the next one? Okay. I have to cover adiponectin. Where does adiponectin come from? Let's go back a couple of slides. Huh? Where is it? Where is it? Can't see it. Where's the adiponectin come from? Where's it come from? Which cells produce it? Fat cells. So, there it is there. Adiponectin comes from fat cells. What does it say? It says, my fat cells, bless you, have less storage. What does it do? It gets back to your brain. So from your fat cells, just imagine that. Fat cells producing adiponectin back to your brain. I'm almost empty, go and fuel up. Back to the brain to do that. Okay, let's go and do that. We done with this one? Yeah, okay. Let's go and do that. But we're going to change all of the key enzyme regulators, all the intermediates. You write first, and I'll start talking about this slide. But as you write, notice all of our acronyms up there. What do you see? PFK2, GLUT transporters, fatty acid synthase, hormone-sensitive lipase, this thing, HMGR, GPAT, GS, elongation factor 2, and mTOR. Make sure you circle mTOR, a major transcriptional regulator. These guys are responding to adiponectin coming from your fat cells. They don't just go to your brain. Write that down. Not just to your brain, they go to other cells, other tissues. And in those other cells and tissues, they change their overall metabolism balance. That's why this schematic looks a bit like a balancing act. So what's adiponectin telling cells to do? Tell your brain to do? Eat more. Tells you your fat cells are running low. Fuel up. Now, while you're writing, and you want to write this down a few times, your body, I said at the beginning of your semester, is generally the rough number of fat cells that it's going to have for the most of its life until you induce cell division. And your body tries to maintain, I say tries to maintain, that overall balance for most of your life post-pubertally, except for disease states, pregnancy states, but it's common. As you age, it starts to decline more, and you see that in very aged individuals. We ready to talk about this one? Okay, there's your fat cell producing adiponectin, particularly during fasting. The longer you fast, the more adiponectin. Write that down. You starve for longer, because next week we're going to talk about starving for days and weeks and what happens to your blood glucose levels. What does adiponectin do? It charges up this AMP kinase. You'll notice exercise tends to do the same, doesn't it? Exercise is stimulating muscle accretion, laying down protein, making more mitochondria, making more cell machinery to do the work. That's the endurance training we talked a bit about earlier. What does AMP kinase do? Well, it increases PFK2, that's the green triangle. It increases the number of glucose transporters, so just before you wake up, if you've been exercising, you kind of get really little bit jittery, ready to go, go for a run, get something to eat. You're more prepared for activity, like we saw at the beginning of today. That kind of makes sense? Glucose transporters, you get them primed, particularly on muscle cells, ready for work, as always. Liver's going to be busy doing its own thing. What about fatty acid oxidation? Well, fatty acid synthase, you tend to say, well, I'm going to oxidize. Fatty acid synthesis, I'm going to decrease that. That's what you're doing in this process. Lipolysis, you're decreasing. The hormone-sensitive lipase, remember that character? HSL activates fat, triacylglycerols. We saw perilipin and the like. That's going to be altered as well. Cholesterol and isoprenoid synthesis. I'll come back to HMGR next week. I'll tell you what that acronym is. You might want to look it up in the meantime. Triacyl glycerol synthesis, what are you doing? Reducing that through the GPAT protein. Glycogen synthesis through glycogen synthase, reducing that. Protein synthesis, you're reducing that through this process, but it's elongation factors. The EEF means eukaryotic elongation factors. mTOR is a master transcriptional regulator I'm going to touch on today to get you ready for. What does it do down here in your heart? It increases fatty acid oxidation, glucose uptake, glycolysis, skeletal muscle, so that's striated muscle, not smooth muscle. Fatty acid oxidation is increased, glucose uptake's increased. What does it tell your brain? You'll notice feeding behaviour increases. And I want you to circle energy expenditure, the signal to go and do work, that's decreasing. But your brain is becoming more active at problem solving, at getting the food. Pancreatic beta cells, you produce less insulin, liver, less fats, less cholesterol, fat cells, less fat acid synthesis and less lipolysis. This protects you, keeping your fat cells just a little bit lean, so to speak, a little less full, makes organs sensitive to insulin, protects it against atherosclerosis, inflammation. Underline those two. We're going to talk about foaming macrophages next week. We're going to talk about the effects of too much fat buildup around cells, attracting immune cells to do damage on your tissues as we run through this process. Please note, like everything we've seen, we've got receptors on cell surfaces, including neuronal cells in your brain. So I'm getting a signal up here. From adiponectin, what's the signal I'm getting? Go back here. Think about the fed, fasted for long periods, and stress state. Is a fasting for a long period a stress state? It is indeed. You're pushing the system beyond a normal diurnal cycle. Keep that in mind. Oops, we done with this one? Okay. Now, what I've tried to do on this schema, I've modified a little bit just to help you with the notes. There are a number of other hormones that are out there, some crazier acronyms doing specific jobs. These are the key ones for this unit. You'll notice here things like emotional stress, perceived stress, cognitive stress. I believe it's a stress, it's not physically a stress. It's not being burnt, it's not being frozen, it's not being having my arm ripped off, for example. That's a perceived stress which then alters your brain function. We've seen insulin and glucagon. Where have we got those from? We should also include somatostatin, the islet cells, the alpha, beta and gamma cells. Cholecystokinin. Now, unfortunately, like a lot of hormones, it's not just one. There's usually a handful and what we call family of hormones. CCK comes from a family of hormones. CCK itself is usually between 8 and 58 amino acids long. It stimulates the release of digestive enzymes. So when we have ghrelin saying you're hungry, you know the rumbling sensation? That's this guy, CCK getting all the acid. So what's CCK doing to you if I put the buffet out the front and you're looking at it for 2 hours? It's getting your stomach primed, lots of acid juices ready to go. So as soon as I eat, what can I do? Process it. Does that kind of make sense? You're priming, like I said for the beginning of today. Once it's primed and everything's working, what do? You activate bile. What did bile do? It's a detergent for fats, isn't it? Separates them, breaks them up, emulsifies them so you can cleave them and absorb them. Can act as an appetite suppressor. Because I've got acid coming in, it's filling up my stomach, getting ready for ingested food. It's saying, well, I can see the food's coming in, now I can start to inhibit that hunger sensation. Ghrelin, we saw from our gut as well, orexigenic. Somatostatin, I've talked about before, another peptide, and there's actually a family member of those characters as well. Produced by our which cells in the pancreas? Islets, alpha, beta or gamma? Gamma. What do? Well, somatostatins affect the neurons, the cell proliferation, inhibiting release of neurons, secondary hormone including insulin and glucagon. So they feed back on that as well. Run around here to peptide YY. 36 amino acids produced by the ileum and colon. I'm going to summarise this for you as well. In response to feeding, inhibits appetite. Once the food's in there, my intestines swell up a bit, it says you don't need to take any more in. So if you are on one of those binge eating hot dog competitions, it's like, well, I've just overridden my peptide YY signal from my gut. Keep in mind, down here, that depends on how much fast fluid flow, how quickly your intestines work. Some of you know that if you eat something, it kind of passes through pretty quick, doesn't it? You've all had asparagus at some point? Yes? Immediately you ingest asparagus next time, as soon as it goes in your mouth, go to the toilet. See if you can smell asparagus in your urine. That's how quickly it gets in. That gives you an idea of the rate of passage. Now, for your rate of passage of food, it depends on the nature of the food, how much you're exercising, what else is going in. It can be a few hours to 24 hours, depending on what you're experiencing. And you know, if you've all had diarrhea at some point, it can come through pretty quickly then, can't it? That changes with your foodstuffs. That is altering motility, those smooth muscle contractions that get things pumping through. We've just seen adiponectin and leptin. They are anorexigenic. Anorexic, anorexic, anorexigenic. Similar terms. Gastrin's another one. Peptide produced in your stomach, duodenum. What's that about? Just circle HCl. But it also alters gut flow. Movement from the stomach into the next stage. T3 and T4, what do? They alter basal metabolic rate. They either upregulate, in most cases, not enough of them, they tend to downregulate. But they're also growth factors, because if I have lots of energy, and I need you to jot this down now, if I have lots of energy, I can do more work, I can make more things. I can make more proteins, for example. I can repair tissue. If I am fasting during my adolescent growth phase, what happens to me? I do not grow as quickly. That's why when you were a newborn in this country, you had the midwife come around checking on your growth rate over the first few months to make sure you're on track at the right growth rate. You should be checked on that. And as you age, you're on a growth decline. Neuropeptide Y, vasoconstrictor, does other things as well. These hormones don't just do one job, they do multiple jobs. But all of them have receptors, signal transduction, they alter activity in the cell, and what you're going to jot down is your cells have all of their receptors on there all the time. It's depending on how responsive they are, and just like you with the kitchen and your phone, your cells are integrating that information to decide which signal is going to dominate, how the cell's going to respond. Does that kind of make sense? What you do, what you eat, when you eat, based on diurnal rhythm, based on how much you eat, your normal activity, alters what you do. What you find is tasteful to eat. Can I get to the next one? Okay. You should know these basic ones. There are many more. This is a basic table you should get to know well. What you're going to do is underline where my laser is here. You'll see that little gap there. What have you got? Peptides. Catecholamines, what are they? Adrenaline, derived from amino acid, very labile. Eicosanoids, where are they derived from? Fats, fatty acids, good job. They are made when you need them. You're going to underline those. What do they have? Plasma membrane receptors and a lot of second messengers. Just circle eicosanoids. Remember, we're going to see those mostly in immune defence. Underneath that line, you're going to write down here or underline it, nuclear receptors. What have you got here? Steroids, including cortisol, estradiol, aldosterone, vitamin D, retinoid, thyroid hormones like T3. Those guys use nuclear receptors. Like we saw with that flick knife for cortisol getting into the nucleus. What do? They alter transcription. They alter gene expression. They're not going to mess with the cytoplasm, they're going to get and make more machinery, if that's the case. The last one, haven't we seen nitric oxide before? Didn't I say that was an unusual molecule? Think of those characters in those cars that go really fast with nitric oxide, the NOx gas. Well, think of nitric oxide as unique. It is a gas, nitrogen and oxygen. It is a cellular messenger. We see next semester. What does it do? From arginine and oxygen, it's a cytosolic receptor. Smooth muscle is involved in its response. Smooth muscle for blood vessels increase with nitrogen oxide. Changes the behaviour of smooth muscles. You should know this table well enough to know which ones use cell surface receptors, which ones use nuclear receptors. And the odd person out, so to speak, is nitric oxide. Yes, ma'am.
So just going back to the last slide, I'm a bit confused about how adiponectin is an anorexigenic,
Yep,
but I thought it was when your fat cells shrink.
yep. Be patient, we're coming to all that, if you don't mind, yep. Because we're going to see what happens in your brain shortly. Yep. Just about done? Now, before I go on, it might be worthwhile keeping in mind, anorexia and obesity are not simple things to solve. And indeed, diabetes is not a simple disease to solve and resolve. They are very complex disorders. You can start to see, hopefully now, why they're potentially so complex. There's so many inputs to what's going on. And as I just mentioned a moment ago, we're going to start seeing what happens at the brain level in a minute. And this is only the simple stuff. I've distilled all this down to give you an idea of how much interaction goes on in the brain. Can I move to the next one? Is that a yes? Okay. What I'd like you to do is you write first, and I'll have another break. Grab something to eat. Ready to talk about this one? Right. If you thought the last lot was complex, we're about to just make it another level up. What I'd like you to do is just draw around this box here that says hypothalamus. These blobby red things and green things are nerve cells, neuronal cells. They secrete a number of things. They're responding to the external environment from different inputs. And here's one example I'm going to get you to think about. This controls eating in the brain. And what you see above that hypothalamus is a seesaw with you taking in a couple of burgers, but that flame means you're exercising, you're burning, you're working, you are generating energy activity. The balance in this process means that you eat less, burn more. We go backwards, who's inducing that? Well, in your hypothalamus, that's these neurons, which are termed second-order neurons, that are telling your hypothalamus to say, let's get you eating less, burning more. Who stimulates that neuronal cell? Well, the arcuate nucleus, which is back up in your brain. That arcuate nucleus produces this molecule, and we've seen alpha MSH before. Yes, it's involved in moving melanin vesicles around your cells, but in your brain, these arcuate neurons produce alpha melanocyte stimulating hormone. That tells the second orders to say, eat less, burn more. It's changing your activity on top of your diurnal rhythm. Where's alpha MSH come from? Well, I asked you right at the beginning to write down pro-opiomelanocortin. Now you can see the spelling. POMC is a big gene, produces a nice big mRNA. From that, you produce this long colour-coded peptide, amino carboxy, with a signal sequence at the front, which means we're going to make it, put it into the ER. We're going to post-translationally modify it. We're going to post-translationally modify it, and we're going to clip out different fragments. And you'll notice they are colour-coded. You'll see the kind of purpley, light purple, dark purple here. That is one fragment's coming out as ACTH. You're going to write down there 1 to 39. It turns out that amino acid 1 to 24 is what does the job, but that you secrete from your pituitary is 1 to 39. In your pituitary, you've got vesicles of ACTH lined up ready for secretion when you think you've got to dump out that stress response. But also in your pituitary, you've cut some of that and packaged it up into alpha-SMHH vesicles and CLP vesicles, clip vesicles as well. From that same POMC peptide, you produced gamma melanocyte stimulating hormone, and these other characters, beta-lipotrophin, which produces beta-MSH, and beta-endorphin. We're talking about endorphins just before we started. What does an endorphin make you feel like? Sorry? Feel good. I know when I go for a really long run, come back, yes, I might be a bit ketogenic, but brain feels really cool, like you're on a high. That's the endorphin rush. People get it for different reasons, particularly from drugs. So what we've got are these characters, then that endorphin produces metenkephalin. They're all vesicles storing these hormones. This is just one long peptide cut into fragments, stored in vesicles to release when there's a particular response stimulus. Where's alpha MSH? Well, there it is there. That same peptide is produced in these arcuate nucleus neuronal cells. What does it tell your cells? Eat less, metabolize more. Hormone, receptor. It's not time to sleep. Hormone receptor. Now you have to be awake again. Hormone receptor signal transduction, and not just in tissues now in your whole body, it's actually in your brain. In your brain, you're getting the signal, I'm not hungry. Other issues might be, yes, I am hungry. Maybe my fat cells aren't quite as full as they should be, but my brain's getting a signal. So what you're doing is training your brain now to the neuronal signal in your brain. You need to distinguish the two. Normally you get feedback to the brain. Here what you're seeing is, well, if I have lots of alpha MSH, I can overcome whatever else is going on, eat less, burn more. If I look over to the right, I see that neuropeptide Y again, that character. What does it do? It comes from these arcuate nucleus cells, neuronal cells, and it tells my second orders, eat more, metabolize less. That is, eat more burgers, hot dogs, don't exercise. So whatever else my body's doing, my fat cells, I've got a peptide, NPY, that says trough out and sloth out. All of these have interactions. So you see the green triangle says let's go forward and eat more, but you start to see the red crosses crisscrossing like some crazy driver saying, well, other molecules like leptin tells my alpha MSH to get busy. Leptin tells neuropeptide Y, producing neurons, don't get busy. Insulin says neuropeptide Y, don't get busy either. Why? Not neuropeptide Y, but why? Why is that? Sorry? Yeah. Down here. So that's your adiposity signals, what your fat cells and other things are doing about whether you're storing stuff. Peptide YY, we've already seen that, GLP from your gut, they're satiety signals. I'm satiated. I'm full. I don't need any more. Ghrelin, that's the hunger signal. So whatever's going on in my body, these hormones are coming out telling my brain either slow down, don't eat so much, or eat more. But I want you to notice the seesaw. If I eat less, what am I doing? Generally burning more. If I eat more, what am I doing? Generally burning less. So I tend to see that balancing act in my brain as well. Excuse me. And this is where it becomes an issue, because in your brain, these hormones essentially give you good feelings, like endorphins. A meal can taste good, but not only taste good, it's addictive. So you want to eat more when you don't necessarily need to. You comfortable with this concept? Okay, can we get to the next one? All right. Now, I need you to look at the green box. Just note that's the hypothalamus again. Where's the hypothalamus? Base of your brain, integrating all the signals, neuronal to hormonal. What have we got in here? Well, we've got some regions, and we've got these hormones, for example, a whole bunch of them. Now, what I've done here is put in some references for you. They're a little bit older than I'd like, but they're very good summaries of what goes on for the process of addiction behaviour for food, why people do become addicted to food, rather than listening to their signals from other tissues, like fat cells, like the pancreas, like your gut, like your stomach particularly. All of these send out hormones like leptin, insulin, CCK, protein Y, ghrelin. They stimulate the hypothalamus, producing this kind of molecule, neuropeptide Y, and this thing, AGRP. Sorry, I didn't name it. It actually was found in a guinea pig called the agouti guinea pig. That's why it's called the agouti-related protein, expressed in the brain to produce POMC. Didn't we see that a moment ago? Pro-opiomelanocortin. And then this guy, CART. Well, what's CART? If you look down here, cocaine and amphetamine regulated transcript. Let me repeat that. CART is the transcript in your brain that's related to cocaine and amphetamine drug addiction. The same process for food as you see for drugs, that is, slightly illegal ones. In the reward for feeding, for stress, and as a psychostimulant. So food can make you not only feel good, it makes your brain feel like, yes, that's really nice. Not only have I got a glucose or a sugar rush, but I'm using the same process that we see here. So what we see as we run through this cycle, either inhibition or enhancement through this orexin, and please note orexin or hypocretin, mentioned at the beginning, a neuropeptide that regulates arousal, wakefulness, activity, and appetite. Now ask yourself, when you've seen all those docos on drug addicts and the other effects, do you generally see drug addicts, depending on the nature of the drug, do they generally eat more or they generally eat less? Generally less. Because this is the stimulation of their brain. Yes, they might be hardwired, going crazy on ice or something else, hyperactivity, but they generally eat less. So let's go back one slide. What are they doing? Eating less, burning more. That's what's going on in their brains. Through this paraventricular nucleus, through CRF, corticotrophin releasing factor, CRF stimulates more POMC, more ACTH. What do? You alter your food intake. Where does this come from? Your brain, the base of your brain that connects to your pituitary to tell you what to do. Feedback from other sources might be altered. And we come back to this guy, melanin concentrating hormone. What was melanin doing? Melatonin. Altered to day-night cycles, your periodicity in terms of diurnal rhythm. So the sleep-wake cycle, when you're more active, when you're resting and recovering, all of those are coming back to bite you. That's why this lecture looks complicated, but it's actually one big circular argument. Back to diurnal rhythm. I'd strongly encourage you, take the time to read those references. Not for me, for metabolism, there's no questions on it. Take the time to read them and understand it for yourself. Understanding why people who are either obese, maybe with diabetes or are drug addicted, what it does to their metabolism and how you can try and work with them to help them. Can I move to the next one? Okay. In this process of appetite modulation, it's not just linked to what's going on in your brain, whether you eat more or less. It comes back to your fat cells and your muscle cells, what you do with them. Leptin we've already seen before. That's about a 16 kilodalton protein produced from fat cells. Now you're going to underline placenta. Not everybody has a placenta, do they? And placenta gets bigger during pregnancy. Why would it get bigger during pregnancy and produce more leptin? Because you're basically feeding two, aren't you, or more. It has a number of receptors. So it's not just one now, there's half a dozen of receptors it's going to target. It's a satiety hormone, regulating the amount of fat storage by altering the sensation of hunger and energy expenditures. So whether you feel hungry, whether you go and get something to eat, and what you do with that. Here's a nice little schematic to help you out. There's our fat cell producing leptin. What does it do to food intake? What does it do to energy expenditure? That's that seesaw we saw before. How does it generally work? Well, here's a fat cell producing lots of leptin. There's a neuronal signal from your brain. Look at my laser. That's a neuron. Got an axon and a dendrite. There we have releasing norepinephrine. We've seen all semester adrenaline, haven't we? You're going to write down that noradrenaline. Noradrenaline coming from a neuronal cell, altering the noradrenaline receptor called beta-3AR. What was the adrenaline receptor's acronym? I should pop that in as a question in the quiz, shouldn't I? What was the adrenaline receptor's acronym? Beta-3 is for noradrenaline. Beta-1's involved in other things. So it might be beta-2AR. Beta-3AR works through a G protein, GS-alpha we've seen before, our old mate adenyl cyclase, write down capital AC. What does it do? Alter cyclic AMP we've seen before, then protein kinase A. It tells this cell, what is this cell? If it's got big lipid droplets in it. Tells this cell to activate gene expression. What's the gene we're going to activate? What does UCP1 do? What's its other name? We're talking about brown fat now. UCP1's other name. I'm not going to give you a hint. Sorry? Good job. Thermogenin. Someone saved you. So now beta-3AR is being stimulated via our colleague leptin, stimulating G protein, altering UCP1. So what are my fat cells doing if I'm putting more UCP1 into mitochondria? Producing more heat energy. So while you are cold, while you get stimulation, while you might be busy, your mitochondria from your brown fat is producing more heat energy. You are burning fat not just by exercise, you're going to write down you're burning fat by heat production. Stimulated from your brain because leptin told your neurons to produce noradrenaline to activate a signal transduction cascade. So it all comes back, doesn't it? UCP1's back again. Can I get to the next one? Tough day, isn't it, the office? Now, these two slides I'm going to scoot through, but I want you to look big picture. Don't write anything, just look big picture. These things, remember I saw cortisol, here it comes, latches onto a polypeptide, opens up and goes into the nucleus, so it's a cytoplasmic receptor. This cytoplasmic receptor gets into cells and alters gene expression. Don't write anything, we're going to come back to this. The concept here is this ligand, who the hell is it? Well, if I have lots of fatty acids running around in my blood circulation because I've dumped a lot out, my muscle will use some. A bit dangerous, they're going to be grabbed by proteins, held up, maybe BSA for example, but some of them can get into cells. They get into cells and bind to these proteins, these PPARs. What are these? Peroxisome proliferative activated receptors. What the hell's a peroxisome? It's involved in fat metabolism. What's going on here? And again, I don't want you to write. I want you to just think for a change. I'm going to show these slides next week and then the week after. What you've got is a family of these pink, brown things and yellow things that integrate at the receptor level inside the cell to alter gene expression of what's going on in these cells. And this cell will basically determine the responsiveness, these receptors, the responsiveness of this tissue as you go through the process. Why? Well, in a muscle cell, you're mostly using fat for work, aren't you? But you have to balance out that activity, whether I work hard, burn more fat, or whether I'm just slothing around watching TV or your computers. Keep that in mind as we go into the next couple of weeks. Come back to it. And this one I'm going to come back to as well. Don't write. Remember I said mTOR was a key transcriptional regulator? It's not the only one, but it's central. Don't write, just think about it. If I'm going through growth, what do I need? Genes to make things. Lots of them. More energy, more activity. If I'm not going through growth, I need less of those. So why not have some kind of general master controller, which is what you see here. mTOR, what does it do? Those little things sticking out there says it gets busy. Don't write anything down. It gets busy because I'm ingesting a lot of stuff. In comes food. What I'm also getting is signals during the growth phase, lots of growth hormones, growth stimulators. What do? Tells mTOR to get busy. mTOR gets busy, and what does it do? Well, it tells glycolysis, get busy. But if I'm getting busy making things, making more cells, what do I need? Nucleotides, more pentoses, hence the pentose phosphate pathway. There's my nucleotides. If I'm getting busy making things, what do I need? More ribosomes. Don't write, just relax. Think of the concept. I need more ribosomes. Don't write, just think of the concept. If I'm going to make more messenger RNA polypeptides, what do I need? Ribosomes. What about that? I need to make more proteins. If I need more cells, I need more membranes, more cytoplasmic, more nuclei, more Golgi, etc. All of those activate these characters. And I'm going to come back to them, as I said, because these guys are driven by transcriptional regulators like testosterone, cortisol, aldosterone, estradiol, etc. The bit I want you to walk away with is that it's not a kinase now that's going to phosphorylate tyrosine, it's a serine threonine. Tyrosine with kinase we've seen for activity around the cytoplasm. The serine threonine is kind of unique for telling everybody to get busy through mTOR. Does that kind of make sense?